Hi, this is Bob Lessick at Johns Hopkins. This short lecture will cover viruses, particularly viral genomes. We'll look at three types of genomes and how influenza subtypes are determined. Our first objective is to examine three broad categories of genome types. Some viruses have an RNA genome, others have DNA. Another category uses an RNA genome, but first uses reverse transcriptase to make a DNA intermediate during its replication cycle. Those are called retroviruses. We'll take a look at a retroviral genome. They are small with only a few genes. In the context of influenza, we will examine hemagglutinin and neuraminidase, two viral proteins. And those viral proteins help describe the influenza subtype. You may have heard of these subtypes. Finally, we will briefly mention the NCBI influenza resource. It's a large collection of influenza sequences and data. Perhaps the simplest viral genomes are RNA-based. The RNA is used to be translated into viral proteins. And it is also replicated using a polymerase that takes an RNA template and makes an RNA strand that is complementary to the template. This has to be done twice because the first RNA synthesized is complementary RNA, which then must be used as a template to make a copy of the original RNA genome. Other viruses have DNA genomes. Upon infecting a cell, the DNA genome must be transcribed so that viral proteins can be translated. That DNA must also be replicated into new viral genomes. Retroviruses have viral genomes, but there is a twist. The RNA genome can be translated into viral proteins. However, its replication cycle involves the use of reverse transcriptase. Well, we know that DNA can be transcribed into RNA. When a DNA molecule is made based on an RNA template, then that is called reverse transcription. That DNA intermediate can be transcribed to synthesize new viral genomes. We can divide RNA viruses into three broad classifications. Some are actually double-stranded like DNA. RNA can form a double-stranded molecule. Of the single-stranded, there are two types. Positive or plus-strand or sense RNA viruses are ones where viral proteins can be translated directly from the viral genome. There are many, including SARS and polio. Others have negative strand or antisense RNA. Before translation, an RNA complement of the genome must be synthesized so that translation of viral proteins can occur. RNA has a replication cycle that is far more error-prone than a DNA replication cycle. That's why DNA is better suited for genome use in most organisms. So if RNA viruses can evolve quickly, that makes finding vaccines very challenging. As for DNA viruses, a large majority use double-stranded DNA. Some use single-stranded. Most of those are bacteriophages or viruses that infect bacteria. Their challenge is to replicate the genome using cellular machinery and to transcribe RNA to be translated into viral proteins. Here are some examples. Most DNA viruses evolve slowly. Retroviruses have what seems to be a complicated cycle. There is a DNA intermediate in replication that adds some stability to the process. That DNA intermediate finds its way into the host genome and can sit passively in what could be called a latent phase. The term enveloped virus refers to the outside of the virus itself. If it's lipid-based, like a cell membrane, it's an enveloped virus. Many viruses simply have a protein coat without lipids. Some examples include HIV, the human immunodeficiency virus, and hepatitis B. Many retroviruses have been linked to cancer risk. The basic retroviral genome has only 3 genes. Often, the protein-gene products are cleaved into smaller proteins. GAG is the group-specific antigen, which makes proteins that are needed for the interior of the virus. Pol is the polymerase. For retroviruses, that is reverse transcriptase. The retrovirus carries the gene it needs for its own replication cycle. Sometimes the protease activity for cleaving the translated proteins into smaller proteins is found in the Pol gene. ENV stands for envelope, and those are the proteins that integrate into the lipid-based envelope. Here are some examples. From the simple murine leukemia virus at the top, there are 3 genes. With some other retroviruses, you can see the smaller protein products within the gene. For instance, in HTLV1, human T-cell leukemia virus, the GAG protein is cleaved into P19, P24, and P15. The terminology is based on protein size. P19 is 19 kilodaltons, for instance. But basically, they all have some form of GAG, Pol, and ENV genes, and maybe some additional smaller ones. Note that RT means reverse transcriptase, and PR means protease. Maybe you can figure out or look up some of the other abbreviations if you're interested. Here is a different virus entirely. Influenza is negative strand antisense RNA, but with 8 segments. We'll focus a little more on 2 of them. Segment 4 is HA, or hemagglutinin, and section 6 is NA, or neuraminidase. Hemagglutinin is a surface glycoprotein. So what does that mean? A glycoprotein is a protein with sugar molecules attached. Surface means it's found on the outside of the virus itself. It's used to bind sialic acids in cell membranes that the virus will infect. Now, there are 18 known types that we can number from H1 to H18. Neuraminidase is an enzyme that breaks down sialic acids. It protrudes in a mushroom-like shape from the viral surface, making it a good target for the immune system. Neuraminidase inhibitors have been used as antiviral drugs. The 9 known influenza subtypes can be numbered N1 through N9. Now we can decode a few things. You've probably heard of H1N1. That caused a devastating flu in 1918 and a bit of a panic in 2009. What exactly is H1N1? Well, influenza A subtypes are determined by the type of hemagglutinin and the type of neuraminidase. H1N1 has the type 1 HA gene and the type 1 NA gene. Now, if you're working on vaccines, it's very important to survey and predict which subtypes might be prevalent in any given winter, whether it's northern or southern hemisphere. I'm simply going to show you the address to the NCBI influenza resource and mention that there's a lot of data there. That is very useful to influenza researchers. One thing you can do is some geographic and time studies to show how viruses from certain places, times, and subtypes are related to each other. Note that there is sequence variation, even within subtypes. To wrap up our really short look at viruses, they have very small genomes, even compared to bacteria. Some mutate quickly, especially RNA viruses and retroviruses. The retroviral genome is really simple, usually 3 genes. And in flu viruses, hemagglutinin and neuraminidase are used to determine subtypes. That's really important for vaccine purposes. Take some time to check the web and NCBI for more on viruses. Good luck.